Good morning, good afternoon to everyone who joins this symposium, and thanks to Valentina for organizing it and for inviting me to participate. I'll be talking about traceable and transparent experimental records, and I'm Anton Bespalov with a Partnership for Assessment and Accreditation of Scientific Practice in Germany, in Heidelberg. So I need to make, for transparency, a couple of disclosures. I'm a co-founder and managing director of PASP, which is a commercial entity, as well as a biotech company called Exciva. And I'm also co-founder of a non-profit foundation called Go Equipped. So one of the largest and hopefully most impactful projects that we were involved in was Equipped, so enhancing quality in preclinical data. This was an IMI-funded collaboration of 29 partners from 8 countries, and as well as more than 100 different associated partners, stakeholders, and advisors. So the project ran from 2017 to 2021. And then to set the path for sustained future of Equipped and the products it developed, we founded a non-profit organization called Go Equipped. So if someone is interested to learn more about Go Equipped and what it does, then you can use a QR code here on the screen. So one of the main projects, one of the core deliverables that Equipped project had was a novel quality system that was designed to support all areas of non-regulated biomedical research. I know, of course, that for many people involved in non-regulated research, whether it's basic, applied, or translational, quality system sounds foreign, burdensome, and likely unnecessary. I will therefore take a minute to explain what the Equipped quality system is about. We formulated 5 key principles that describe the essence of the Equipped quality system. And 2 most relevant principles for our discussion today are, of course, that key research processes must be transparent and traceable. It may all sound too complicated, too difficult, so let me just immediately jump to one example. Just to make it really simple. Equipped, for example, strongly recommends applying randomization in your study design, but it is up to you to decide whether, up to scientists to decide whether you apply randomization and so when you apply it, and decision, but the decision as well as all essential details. Actually, sorry, it blocks my screen, part of it. Sorry. But the decision and the all essential details, so methods used for randomization, must be recorded and must be traceable. So it doesn't really matter whether we are talking about randomization or about any other aspect of study design or data management. Please note that here we really make an emphasis on transparency and traceability of all decisions and all methodological details. So let me take another example. In a survey that was very commonly, broadly discussed about a few years ago, a survey that was published by journal Nature in 2016, a large number of researchers were asked about So they were asked various questions regarding to reproducibility of their own experiments, as well as experiments reported by their peers. So one of the questions was, have you failed to reproduce an experiment? And as the results shown on this slide illustrates that across all areas, it's not only in biology, more than 40% of survey participants experienced failure to reproduce their own experiments, and the failure rate was even greater for experiments that were conducted by others. So results of this survey are frequently cited in the context of the general debate on reproducibility of research data. Sometimes people even talk about reproducibility crisis, but we view these results, however, from somewhat different angle. So very often when people talk about reproducibility of data, it is actually about quality of experimental records. So now it's time really to talk what are the experimental records. And these are essentially traces that our research activities leave. So we leave traces all the time. So when we shop, when we meet with friends, we travel, we may or may not keep the receipts for the goods we buy, or we may or may not keep train tickets from our vacation. But traces left by our research must be retained, and preferably in the original form. So collection of traces in research is called experimental records. Experimental record includes all information from study planning, study execution, study analysis, and may cover more than one experiment that belongs to one study. It includes study protocol, raw data, process data, reports. And as you know, most funders require these days data management plans to be included in the grant applications. But very often data management plans are not exactly what they're supposed to be. And this is why we say that data management is about making sure that experimental records contains everything that you may need later to reconstruct what exactly was done by whom, when, and how. So, but why would we want to reconstruct what was done in the past? Just imagine we need to revisit an experiments conducted by us or by our colleagues. Say we need to travel back in time several months or even 7 years back. So would I know where to search for the information related to this particular experiment? Do I know what to search for? For example, do I know how the folders and files with relevant information are called? If I have an idea where it should be, but is it actually there? Is the needed a needed set of information complete? Is there anything missing? And even if all of the previous questions can be answered with yes, would I be able to repeat the experiment done before, especially if it was done by my colleague who has already left the laboratory? So why would I want to repeat? There is a really there is a terrific example that was published several years ago and illustrates why from time to time we do need to revisit experiments conducted in the past. So in this particular case, two independent laboratories, one in Boston and another at Berkeley, decided to collaborate to establish FACS profiles of primary breast cancer cells. And it came as a surprise to them that when they had a single aligned protocol, still they were generating consistently different outcomes dependent on whether, on which lab performed the experiment. Despite using identical methods and careful validation of all antibodies used, one lab could not reproduce the CD10 and CD44 expression profiles obtained by the other. So tissues that were digested into organoids and processed in Boston always produce the results, so matching the Boston profile, and tissues treated at Berkeley always produced, let's say, Berkeley profile. So it took a long while and a lot of efforts to establish that the difference in the outcome stemmed from two labs using different mechanical shaker speeds to disrupt the tissue. In Boston, the tissue was stirred in a flask with a stir bar more vigorously compared to the Berkeley methods, where tissues were incubated relatively gently on a rotating platform, but for a longer time. As a consequence, from this slow digestion strategy, 5 times more organoids were obtained at Berkeley compared to the fast digestion protocols that apparently had a very dramatic impact on CD44 antigen presentation. So as a stirring speed was considered a insignificant methodological detail, technical detail, not worth reporting. It took those two groups more than 2 years and a lot of additional resources to understand this data reproducibility, quote unquote, issue. So what do we learn from this example? So first, experimental protocols should always include as many details as possible. And second, data management concerns not only research data in the conventional sense, as presented in figures and tables in our publications, but also all metadata, all traces of all traces that our research activities leave. So industry and academia may have different approaches to data management and data integrity, but in the long run, every research organization at some points of time shares the results of its work with peers, collaborators, authorities, or business partners. And in the academic world, a set of principles known as FAIR are actively endorsed by a variety of organizations and initiatives. FAIR stands for data being findable, accessible, interoperable, and reusable. And FAIR focuses heavily on metadata, basically on information describing research data, because because this is what makes sharing with the peers possible. In contrast, industry is guided by the so-called ALCOA principles. And ALCOA is an acronym for attributable, legible, contemporaneous, original, and accurate. One could say that ALCOA focuses on attributes that are directly related to the data and on the experiment itself. So despite differences, FAIR and ALCOA have a common aim. So because we generate data, not because we want to archive it and just forget about it, but we generate data because we have an intent to use data. And this is what both ALCOA and FAIR actually help us to achieve. I guess you will hear today about FAIR after lunch, and I will spend some time now to explain ALCOA principles. So everything we save as a part of experimental records must be attributable throughout the entire chain from study design, materials and samples, data acquisition, data analysis, and up to reporting. So all information we collect must be attributable. If we find an old coin, we may not be able to say immediately to which state and to which ruler this coin should be attributed. We may not even know how old it is. We seek experts and who, after some time and some efforts, and with a certain degree of confidence, not 100%, but certain degree of confidence, will communicate their opinion. So in research, we do or we should do it differently. To find out who is the author of a certain piece of data, we do not launch an investigation. So we do not make phone calls or write emails to those who may have an answer. So we have all required information. So we must have all required information in the experimental record. A working example of this principle is that actually at the end of my presentation, you will find a slide listing sources, source or authors of images and illustrations that I use in the presentation, and so authors to whom those images should be attributed. So the first letter in ALCOA stands for attributable. That is, who acquired data, who performed an action. Basically, it means that the originator of the data must be uniquely identifiable. So there must be a written record of this. So adherence to this principle means that the names or initials of the researchers are added to the records. So in the lab notebook, for example, in file names, folder structures, it can be in various different formats, of course. Second letter stands for legible. That is, can you read and understand the data? This is important because it allows a reconstruction by others interested in your records. One common issue that we have in sometimes related to legibility of records is handwritten notes. We perfectly understand our own writing, but others may not be able to read it. It is also important very often to consider using a common language in the lab. For example, if you have different lab members coming from different countries and speaking different languages, and it may be wise to agree that all lab notebook entries are written in the same language. Potential issues so I will not, of course, be able to cover all the potential issues with legibility, but one thing to mention is that it is often believed that once we transform our experimental records into digital forms, then the legibility issue is solved. However, we should not forget that laboratory equipment often generates output in a format that requires specific hardware or software, or maybe simply not available in a few years when we will need access to our experimental records. Contemporaneous means that the data must be recorded and generated, must be recorded when it is generated, and that no backdating should occur. Therefore, it is best practice to always document immediately and or have dedicated times for creating records, maybe at the end of the day or at a specific reserved time during the week. The letter O stands in ALCOA stands for original. and it indicates that all research data must be saved in the original format, meaning that we do save all information during the conduct of the experiments as it is generated, and in a way that it cannot be, should not be altered. This is especially important for the storage of raw data. And just to make it very clear, so raw data are all original records and documentation, which are a result of the observations and different activities in our studies. And this is not, raw data is typically not what we keep later in Excel spreadsheets on our office computers. Finally, data should also be accurate, meaning that data should be recorded correctly and truthfully. In practice, of course, we know that errors happen, and they have the right to exist and may be overseen. Therefore, it is highly advisable to think about the accuracy of records that we create before the files are closed and the records are made permanent. For example, some organizations implement so-called 4I principle, meaning that at least 2 people see and review important documents. So depending on the structure and the needs of your organization, so accuracy of the experimental records can be secured by, for example, asking a colleague, another student, another postdoc, a technician to do a quality check to make sure that nothing is missing or and the record is accurate. So, is the data management burden? Yes, of course. If you see, if you look at this long list of expanded Alcoa items, we covered only 5 of the attributes. Yes, of course, it is a burden if we do not understand what it means and why we need it. When we work with the labs to help them improve data management practices, we often perform a very simple exercise. So we ask to identify any table or a figure from a recent publication or from not so recent publication, and really randomly pick up some set of data. For instance, it could be a bar graph such as what you see here on the slide, and then we perform a traceability experiment. So we ask scientists to go back to the original raw data. Could be in a paper-based lab notebook, could be in an electronic lab notebook. But what we are interested in is how whether scientists are able to trace back how reported data how raw data were processed and what kind of analysis was performed, and eventually how they appeared in the final form in a publication. And you'll be surprised that this is not always an easy exercise. Try to do it yourself with the papers that you published 1 or 2 or 5 years ago. In all research environments, decisions are constantly made at different levels, whether we want to develop a project in one or another direction, whether results are solid enough to be shared with our peers, or maybe we need to make a decision whether first the investment of time and efforts is justified by results that are obtained so far. These decisions are often shared and discussed with colleagues, authorities, and other peers. And if the decisions are based on incorrect or incomplete data, then this will ultimately result in loss of time, efforts, money. For example, you come to a new lab and want to build on a project from a previous student or postdoc, and you certainly don't want to waste time simply because previous experiments were not properly documented. Another common concern is the ethical use of resources. So our society grants us a privilege to do research using taxpayers' money, accepts or may accept our arguments about the use of ethically sensitive resources such as laboratory animals. And in exchange, we owe the society to maintain adequate records of what was done and how it was done. So last but not least, maintaining a high degree of data integrity essentially is a way to protect our reputation. So research data that we produce is our legacy in the lab and for the entire research community if we publish it. Therefore, by ensuring data integrity, we build our reputation and make sure that we do not lose it. So what I was trying to say is that it is, of course, data management is a burden if we don't understand what it means and why we need it. And if we don't do it properly, then we must be prepared to live with the consequences. And let me just take one example. So let's just say that you have a postdoc working in your group who validates a new target X for a certain disease Y, and then a patent application is filed and the project advances based on what you believe is really exciting results. Then what could happen is this postdoc leaves the lab and take some files with him or with her, and so this was the only location of the data were on his or her office computer. Another horror scenario could be that all raw data can be retrieved; they are available, but the original assay conditions are not described. And of course, yet another example is that experiment was actually done several times, but the results that you described in a patent application were obtained only once. Why is this? Why do I say these are horror scenarios? Because from the industry perspective, we need to be aware of the fact that technical reproducibility is one of the cornerstones of patent law. And there are already precedents when people are trying to invalidate intellectual property based on integrity of data records. Another example is related to risks. Well, so any action that's a risk for not just for us, but for our organizations, because any action that we do as individual scientists rarely stay personal without affecting the whole team, the whole working group or projects in general. So one of the most striking, relatively recent examples was from a publicly traded company in the United States that lost half of its market value after inconsistencies or inconsistencies were found in the PhD work of its CEO. So what was interesting in this case, that the stock market responded before any investigation took place to confirm or discard the allegations, and without considering whether observed inconsistencies have any direct link to a company's portfolio projects. So another example, maybe last example, related to risks to our reputation. So this also happened some years ago, in 2016, somewhere in Germany, during a police raid at a research institution, a major research institution. There was identified breaches of animal welfare and drug law, and long story short, after internal investigation, director of the institute was found guilty of research misconduct. Some papers were investigated, and what was found is, maybe beautification of the results. But what was most important, what really got them into trouble, there were no original data or lab books. Again, if you remember what I said earlier, there were, of course, Excel files with data in spreadsheets on office computers, but those were not original records of the experimental records, no original data. So as a result, the paper was retracted, and some corrections were requested in some others. So you can say, well, it's a relatively minor thing, but a public perception in this case was that I don't put the number so that you're not really shocked. But public perception from this case was that there were lots of animal subjects killed unnecessarily. So, and of course, that's also costs head of the institute his reputation. So you may wonder why all of these examples that I use are about risks, as if this were the only way to show you why it is important to follow good research practices. So indeed, classical behavioral theory teaches us that you can efficiently control behavior not only by withholding an aversive stimulus, but also, and it's actually much more efficiently, by means of what is known as positive reinforcement. So that is by presenting an appetitive stimulus made contingent upon certain behavior. Unfortunately, we have very few examples when scientists are rewarded for following best practices. That's why we tell those scary stories. But what happens if there is no contingency between behavior and reinforcement? So again, behavioral theory says that other behaviors may get unintentionally reinforced. And a classical example of is what we know as superstitious behaviors, often prominently displayed in sport. But if we take an example closer to our hearts, what happens if there is no contingency between transparent reporting and reinforcement? In our case, of course, reinforcement could be, and very often is, publication in a journal with a high impact factor. I hope that at least some of you are familiar with the so-called ARRIVE guidelines that were developed to improve reporting of animal experiments. And if you are involved in vivo research, but you do not know those guidelines, please be advised to follow the link in this QR code and learn about it. So according to ARRIVE Essential 10 recommendations, so all publications reporting on the outcome of animal experiments have to provide sufficient information on study design, sample size, estimation, inclusion, exclusion criteria, and so on. So ARRIVE guidelines are endorsed by many journals, and it becomes very common to see scientists state in their publications that ARRIVE guidelines were followed. Please note that the intention of the arrived guidelines was not that we just tick the box by stating that we know the guidelines. The desired behavior is actually reporting according to the guidelines. So we looked at a couple of publications that's acknowledged the RIPE guidelines, and they said that their procedures follow the RIPE guidelines, or at least RIPE guidelines were considered. And as you can see from the summary slide, this is clearly was a box ticking exercise in case of those publications. So why do we do that? In the Nature survey that I mentioned earlier, one of the questions asked was about factors that could boost reproducibility. And it is quite noteworthy that several high-scoring answers pointed at lacking or just deficient training, need of better understanding of statistics, supervision, coaching, and so forth. So there are certain aspects of good research practice that are hardly ever noticed, mentioned. They hardly ever receive attention in the standards academic education curriculum. So the take-home message for today is to make the best use of data, follow the best practices, and to know the best practices and to be able to follow them, invest in training. Do not look for excuses and not to change the convenient routine. If you're interested in learning learning about good research practice, you may have a look at the online course that we have developed and is freely available to anyone. And if you get a copy of my slides, you will also see source data for the images that I used in these slides. And so with that, we'll be happy to answer any questions that you may have.